Okay, so after our last video with the knowledge we've got of DNA, hopefully a little bit of this diagram might make sense from the beginning. We can see the 3' and the 5' ends. So that tells us that this is DNA, also it's a double helix. And if, for example, I put some letters in here, A, T, C, G, G, C, T, A, we can see that complementary base pairing that we saw before. You'll notice something weird is happening here though. We've got this double helix at the top of the diagram, and then it seems to sort of open up. And then down on each end, we've also got what is the beginning of 2 more double helices. And you'll notice that I've also written down the 5' and the 3' ends of these as well. So if I followed the 3 around, you would get to the 5' end, and the same thing on the other way. Okay, so what you can see here is us replicating DNA. And as you can start to see from this diagram, the way that we replicate DNA is by taking this double helix, unzipping it and opening it up, and then reforming it down at the other end. So you end up going from 1 double helix to 2. Now, as you might imagine, when we are replicating DNA and we're creating 2 copies of our original DNA, we want these copies to be exactly the same as our original. So I'm going to draw down this side here some more nitrogen bases, and as I do that, you'll see, if I do a couple just to start off with, you'll see that on the opposite sides I'm going to continue matching up TAATCG. I'm going to continue matching up this the nitrogen bases. You have to imagine that I have just unzipped and started spreading out each side of the DNA, each sugar phosphate backbone, and I'm pulling apart the nitrogen bases that were matching up. So you can see that I've just continued that pattern of adding more nitrogen bases, and you can see that they continue to match up. CG, GC, TA, CG, etc., or the whole way around. So everything matches up perfectly. So we've already understood the first step of this process. Step number 1, DNA has unzipped itself. Step number 2 in this process involves these things. Now these things are free nucleotides. Now free nucleotides are what they sound like. You'll remember before that we drew a nucleotide similar to this with our phosphate, our sugar, and our nitrogen base. Now this sugar phosphate backbone is the same as this black line here, which is what we've got running down the side, and the nitrogen base is what sticks in the middle. So the nucleotides where there's a black line and then a letter on the right hand side would look very much like this. And if there is a black line on the right hand side with a letter, then it would be these kinds of nucleotides. Same thing but just flipped around. So again, sugar phosphate backbone with the nucleotides in the middle. Now these are slightly different to the nucleotides we've got running along here, only because they're just floating around. So in step 2 we're going to see these free floating nucleotides start move towards their complementary pair. So this T will move towards this A, the G towards the C, this C, well actually it would probably try to move up here, and we can see that actually on both sides we've got these free nucleotides connecting up and starting to make the exact same pattern. Now if I continue this process, it'll look something like this. Now you'll notice I've added a note here, step 2, free nucleotides pair with template strand, and that's what we're talking about here, these free nucleotides connecting up. It's the first time I've called it this, the template strand, but the original strand we call a template strand, and I hope it's clear why, because it provides a template for these free nucleotides to then connect up to it. As you can see, I've also continued this this part of the drawing down here, where I've brought in the correct free nucleotides, so A for a T, C for a G, G for a C, and the same thing on the other side. Now the beauty of this process is, as you can see, the template was paired up on either side, but now that we've brought in these free nucleotides, we end up with exactly the same thing. So CG, TA, GC, CG, CG, TA, GC, CG. Because of this template strand being paired up originally, then separated, and because of the fact that you're then bringing in these free nucleotides, you end up with exactly the same nucleotide sequence on both of these new strands. Now we're actually very close to being finished here. One of the last things we need to do is, you'll notice that there are little gaps between each of these nucleotides, and these little lines, remember, are the sugar phosphate backbone. So we need to start connecting up all of these little gaps so that eventually we end up with one straight line. Okay, so that's the third step. And as you can see here, I've written sealed gaps between nucleotides. So those free nucleotides have come in, we've sealed up all of the gaps between all of the sugar phosphate backbones. We now have one straight line. Again, notice this code in the middle with the nitrogen bases is exactly the same on both sides. Finally, step 4, we're going to retwist the double helix, and you can see that happening on both sides. So I hope this whole process makes sense, from unzipping the original DNA double helix, bringing in the free nucleotides and connecting up these new pairs, sealing the gaps, and then retwisting the double helix. Now, there is one more bit of important information that you need to know in order to fully understand this. The process you've now understood, but this unzipping, this bringing in of free nucleotides, reconnecting all of these sugar phosphate backbones and then retwisting, doesn't just happen on its own. And it happens because of enzymes. So there are a number of important enzymes that you need to know about, and I'm going to run through them with you now. So the first step that we talked about was unzipping this double helix. This doesn't just happen on its own, it happens thanks to an enzyme called helicase. Step number 2, free nucleotides pair with template strand. Now this step, where we bring in our free nucleotides and connect them up to their base pairs, this happens because of an enzyme called DNA polymerase. And that's what's doing all of these blue arrows. Finally, we've got steps 3 and 4, where we seal the gaps between the sugar phosphate backbone, and then we retwist the DNA double helix. And that happens thanks to an enzyme called ligase. And that's both steps 3 and 4. Now, those are the most important enzymes for you to know about: helicase, DNA polymerase, and ligase. There are in fact a couple of other ones, and I'm very quickly going to tell you. For some of you this may be more information that you need, but nevertheless I'm going to tell you. And this actually brings in this whole idea of the 3' and the 5' ends. So one thing that's important for you to understand is that DNA polymerase, that's bringing in these free nucleotides, works from the 5' end to the 3' end. So here you can see that this would be working from the 5', this would be the 3' end of this strand, so it would be connecting them up in this order here, which is great. The reason why that's great is because, imagine helicase is like a wedge that will slowly start unzipping this DNA double helix at the top. This is kind of moving in this direction as it's slowly unzipping things, unzipping the DNA double helix, and so you're going to get more of this opening up going on as we go upwards. That works great for this side of my DNA replication, because as this opens up upwards, then my DNA polymerase can just keep joining in my free nucleotides as it goes. So that works great. Unfortunately, on the other side, this strand over here, well, the 5' end is here and the 3' end is here. So actually, it's wanting to try and connect them downwards, even though the whole process is moving upwards. So it's kind of doing it in the wrong direction. The way this side is going to work is it's going to connect up a couple of these nucleotides and create what we call a primer, which is just a few nucleotides, and then there'll be a gap. And what DNA polymerase will do will fill in that gap. And then as this whole process has moved upwards, there'll then be another gap above the original primer that you created. So there'll be another primer that's created, and then DNA polymerase will fill in that gap. In fact, over on this side, the same thing happens. We start off with a primer and then we just work our way upwards. The enzyme that will create these primers is an enzyme called primase. In fact, we don't end up keeping these primers. Another enzyme called exonuclease will come along, remove these primers, and then DNA polymerase will again fill in the gaps. So that's where the 5' and the 3' come in. Because these strands are moving in opposite directions, it actually makes DNA replication slightly more complicated. Easy on one side, slightly harder on the other, because it has to work in little chunks. As I say, the main enzymes that you need to know about: helicase, DNA polymerase, and ligase. The extension of that is then knowing about primase and exonuclease. Okay, well done.